nothing to do with biology. Now we're both in biology. E in plants, at the moment at least, right? Not always. Organization of root cellular patterns is a subject.
Great. Thank you, Christian, and thanks to all the relevant people. So my brief was to talk about plants, and I need to start off with a couple of confessions. So the first is to do with this. So the UK Funding Councils funded six big systems biology centres a few years ago. So the Nottingham one was devoted to plant biology. It was a big operation, highly collaborative, and the upshot of that is I'm perfectly capable of muddling details as we go on, because there's quite a lot of people involved. The second thing is to avoid false pretenses. So these slides, the experimental slides, are all Tony's, the modelling ones are all Nathan's. The other thing, of course, you're told not to start a talk with an apology, so I would apologise for apologising. Right, so that's the Arabidopsis root, which is what the Systems Biology Centre focused on. So Arabidopsis is the model plant species, so the plant equivalent of Drosophila or whatever. One feature of it is the geometry is really clean. It's illustrated on the right. One upshot of that, of course, we've already heard these cell walls are pretty rigid. So in terms of the themes of this program, there's not much to say in terms of cell motility, and there's limited scope for cell morphologies, interesting cell morphologies. So I have chosen the topic most closely related to the third theme of the program, namely pattern formation, which I'll come to in a minute. So this talk is about the formation of vasculature. So there are two parts to it. This is the xylem, and that's the thing I'm going to focus on, the orange bits of the phloem. And there's a description of what they do on the next slide. So the xylem takes the water and some minerals up from the roots. The phloem is to flow in the opposite direction with nutrients. So pattern formation, this is, if you like, a symmetry break from radial symmetry, although given the thing's discrete, you never see strict radial symmetry, but in the early stages of growth, you see patterns that look pretty much radially symmetric. The formation of vasculature does involve destroying that radial symmetry. So here's the vessel with which we're concerned. There's two different shadings here. There's the light blue, which is the xylem poles, which is how the vessel is initiated. And then the darker blue is the xylem axis, which runs all the way across. Yeah.
On the previous one, you had a lot of colored layers. What's the significance of those?
Right, I didn't want to get into that because I'll forget which order they are. It's something like epidermis, cortex, endodermis, pericycle. Have I got that right? In any case, they're different cell types, right? It is a very regular structure, which is one, in mathematical terms, one of the virtues of this thing.
But they're structurally not for transport.
They're not to do with, and the epidermis will play a role later, but they're nothing to do with the vasculature. Yeah. So that's the sort of right. I forgot to say one important thing at the moment. At the beginning, there are two sort of general themes to this talk. One is explicit, and that's to do with pattern formation. So the first half of the talk is what interactions do you need to sustain a pattern in a growing root? And so more generally a growing biological material, but specifically here at the root vasculature. The modelling results I'll present on that are fairly mature. The second part of that pattern forming issue is how does the pattern form in the first place? And the results I'll present on that are rather preliminary. The second general theme will only be implicit, and it's something to do with how far you can get in understanding these kind of systems by generic mathematical studies, and how far you have to take the details of the biology into account. You'll sort of see what I mean by that later on. Anyway, this is one way of breaking down the talk. That, as I say, is the second issue of pattern formation. Where does it come from? That'll be preliminary. This earlier stuff is more mature. Okay, so there are two key plant hormones involved. We've heard quite a lot about auxin in the previous talk, so you can think of these two as morphogens as you like. There's a whiff of Turing patterns in this, but it's not really that. The other thing I want to say straight away is even neighbouring cells are in different states. So this is xylem, this is procambrium. So writing down PDE models here is probably not the way to go, and I will have cell-based models of these things. So the plant hormones are auxin. So where auxin is dominant, you get the xylem, which is the thing we're interested in. So this is a cytokinin mutant, the other hormone. So cytokinin is knocked out and everything's xylem. These are false colours, but they make the point. This is the opposite case. So where cytokinin wins, you get procambium, you get the other state, and that's indicated here in a auxin mutant. So those are the two plant hormones, and I guess the main points here is they're relatively small molecules, so that they are motile. Auxin both diffuses and is transported actively. Cytokinin, as far as we know, is only diffusive. They trigger a whole pile of different gene regulatory networks, and we've heard that alluded to already in terms of auxin. The ones I'll talk about will be rather specific.
Can these molecules cross the cell barriers?
Yeah. So there are these efflux and influx carriers for auxin, as we've heard already, but they also cross the walls. Yeah. They can change charge state and things when they do so. Okay, so auxin is actively transported. So the 2 points about this slide is the transport can take it up the concentration gradient, which will be important. The pin carriers are localized on a particular side of the cell wall, so it is directed transport. This is another picture we basically saw in the previous talk. So the previous talk majored on pin 1. That's not the one of interest to us because it's pretty much uniform across the vasculature. The one I'm going to be concerned with is pin 7, a different type of efflux carrier. And it's the localization up here is the relevant bit because this is where the vasculature is getting formed. These arrows are the auxin flux, so there's this famous fountain effect where it comes down the middle and goes back up the sides. And there for Hans is a record of what the cell layers are called. Oh, I got some of them right. Yeah.
Before you move on.
Yep.
So, did we not just see that the flow in the xylem is up?
This is the auxin flow, not the flow of water.
So, it's my impression that a solute would be carried with the water, so this is really counterintuitive.
Remember, there's these active transporters. You're taking me beyond what I know, but I assume it's the active transporters taking it against the flow. Okay, so as implicit in the previous statements, each of the hormones inhibits the other, but they do it by a very different mechanism. So auxin inhibits cytokinin. The mechanism is not known. It doesn't, as I understand it, it doesn't reduce the level of cytokinin, but stops it being perceived by the relevant response genes. But it is mediated through an intermediate species, which is this AHP thing that I'll come to later. So this is a GFP staining for AHP. So these are the xylem poles, which we'll see again later. So that's the view through the cross section. That's from the side where the AHP is localized. So the mechanism for cytokinin repressing auxin is different because it is one of transport. Actually, I think this is quite interesting in its own right because it's not quite clear if you view it as a repressor or an activator. So it's a repressor in this cell. This cell is cytokinin dominated. The cytokinin expresses these pins, these auxin carriers, which drives the auxin out of the cell. So it depletes the level of auxin in this cell. The activatory bit is in the neighbouring cell. It, of course, correspondingly increases the level of auxin. So this is, again, the experimental demonstration of that. The two pictures on the left are the wild type. So this is GFP stain for pin 7. So the pins are in the procambium region. Here's the xylem in the middle, the auxin-dominated bit, where you don't have the carriers. If you add cytokinin, you mess the whole thing up. So exogenous cytokinin overcomes the auxin in the middle, and you get the uniform pin states. This is the cytokinin mutant, wooden leg. Cytokinin is not functioning here. So again, in the relevant region we're interested in, you're not seeing the pin 7s, you're not seeing the carriers. So the simplest model to think about to describe this, and where I start the mathematical analysis, is the two-cell case. So this is two identical neighbouring cells. This cell has auxin winning, so auxin is at some high level, activates AHP, which represses cytokinin, so cytokinin isn't active. Conversely, in this cell, cytokinin is winning, so it's generating a lot of auxin carriers, which is driving the auxin out and keeping the auxin down here. So these two neighbouring cells are in very different states. Incidentally, there was a comment earlier in the conference that nobody here seemed to be doing gene regulatory networks. We are, and this is the simplest of the ones I'm going to record. Slightly unusually for me, I don't have an enormous number of equations. And those of you who are used to gene regulatory networks won't see anything surprising in these. But I need to, I guess, briefly go through them. So this is the auxin level in cell I. Auxin is transported in and out by two different effects, by diffusion and active transport. So active transport is proportional to the level of pin carriers. That's one important feedback. It's constitutively expressed and it degrades. Cytokinin, similar, except that there's only passive transport, so there's only the diffusive term there. As I go on, I'm going to add more and more ingredients. So at this point, the expression of cytokinin is constitutive, so it's constant. But later on, there's going to be a repressor of cytokinin. So there's, as this concentration increases, you reduce the production rate of cytokinin. In the simplest model, there are only two genes involved. AHP is the one driven by auxin. Pin 7 is the one driven by cytokinin. Cytokinin is driven down by AHP. So as a proxy for that, we drive down pin 7 by AHP. So again, a standard model for transcription with activating terms. So again, this is constitutive production. These are activators. So these species increase the rate of production of the relevant mRNA. These are repressors. It's only on the denominator, so they reduce it. So that's transcription and that's translation in the simplest form. Production rate proportional to mRNA levels and then degradation.
What are those sums over, the J in RI?
Yeah, there's more detail. There's more complexity on this than it deserves. So the H is a Hill coefficient, of course. This is the sum over all the activating species. That's the sum over all the repressing species.
Here you have only one of these.
Yeah, that's why there's too much detail on this slide. Yeah. We do have more species later, but this is the bifurcation structure of that two-cell model. So two identical cells. The two key bifurcation parameters are the two hormone levels. So we tweak those by varying the cytokinin level, and on the next slide, tweaking the auxin level. So at low levels of cytokinin, both cells are in the same state, and that would be viewed as the auxin-dominated case. At very high levels, the two cells are also again in the same state, so in the cytokinin-dominated state. But in between, it's a pitchfork bifurcation because the two cells are identical. One cell will be in the auxin-dominated case with high IHP. The other cell will be in the cytokinin-dominated state with correspondingly low level. And the converse is true here. So this is this branch corresponds to this one, and vice versa. So parameter values, as usual in this kind of model, they are best estimates. Some of them are known reasonably, some you just have to estimate. So it's more an issue of qualitative behavior. I guess one feature is here, if you started off at high cytokinin level, you'd have to drop it an awful lot before you flipped into the pattern state. As I understand it, that there is some experimental evidence for that. It warrants more being done. So the other bifurcation parameter is auxin, and essentially this is the converse picture of the previous one in terms of flipping between states. And in terms of auxin production with these parameter values, this thing is extremely difficult to shift yourself away from if you start off here. You're going to stay in the unpatterned state until you get right down here. Okay, so the upshot of that is that relatively simple mathematical model, rather small number of species, identical ODEs in two different cells, give qualitative behavior consistent with what we want in terms of patterning. But to get something that's more meaningful geometrically, we need to go over to the multi-cell case. So we have a multi-cell model, right? So we do mechanics with this model as well as transport, but this is fixed geometry, so there's going to be no mechanics in it. The cell geometries are real ones, right? So confocals, and then our image analysts extract cell walls from it, and we stick them in the computational model to give the cell geometries. Is there anything else I want to flag from there? So each of the cells has the relevant gene regulatory network in it. There's the transport between cells, both active and diffusive. In some cases, you've got different types of cells in your simulation, so you have to give them different parameter values. So the first set of simulations I'm going to show based on that model. So this again is the issue of how you sustain a pattern. So in this case, we kind of force the pattern by initially putting our pin carriers where the experiments say they should be. But then remember that the pin levels are one of the variables within the model, so they evolve as you run it. So you're not forcing it into the observed state, you're letting it equilibrate from the initial state. So that's where the pins end up. That again is the pin green fluorescent protein. So the business in the middle, the xylem pole, doesn't have the pins, the rest of it does. IA2 is an auxin response gene, so the GUS stain tells you whether that gene is on or not. So where auxin is present, it'll be on. So here you can see the presence of auxin in the relevant xylem axis. So all's well so far, but then there's a slight problem, which leads me to the next part of the talk. So here's AHP along the xylem axis as desired. Cytokinin is everywhere because remember, auxin inactivates cytokinin in terms of the perception of cytokinin, not its level. In the multi-cell model, we have one extra species that I didn't mention when I talked about the simplest level of modeling. We put it there because it's biologically known to be part of this network, so it might as well be there. But really, it's just a proxy for the effect of AHP. The thing that goes wrong is that if you do the GFP stain for AHP, and we've already actually seen that, it is localized at the poles rather than along the entire axis that the modeling predicts. So this is the kind of point where you get slightly cross with biology. Fortunately, in this case, it was already known that there's an extra set of species one might have to worry about, which will explain this. And that's the next thing I'm going to talk about. Okay, so this is earlier results. So this is the wild type cell and where the AHP is localized, as on the previous slide. This is a fabulosa knockout, so it's actually quadruple knockout, so there's a high level of redundancy here, but fabulosa, this thing seems to be the key ingredient, and that's the one we're going to stick in the model. So the point is, when the experimentalists see this picture, it reminds them not of this, but of the right-hand side here, which implies we should be putting PHP into the model in order to get this tighter localization of the AHP. Okay, so fortunately some of the other ingredients to that are also well known. So this is short root and scarecrow, two genes. I think these play a role in root hair formation in various other contexts, so they've appeared in a number of mathematical models. So they're present in the epidermis, in the outer cell layer. They dimerize and cause expression of a microRNA. So the microRNA is able to diffuse inwards from this outer cell layer. It mutually degrades with PHP. So this is the obvious model for that process, and the upshot is equally obvious. mRNA is diffusing in, it's consuming PHP but being consumed at the same time, so it doesn't get all the way in. So you get a kind of radial gradient of PHP as you go into the center of the root. Okay, so putting in that extra subnetwork, as implied here, gives something much more like what we want, with one drawback, and I think this is the least satisfactory part of the initial model. We need a cytokinin inhibitor that actually appeared on a previous slide. And as I understand it, it's not known what this is, but it's probably something very like AHP, which isn't repressed by PHP. So we stick in that inhibitor in the region that AHP has been removed from. And with that choice, you get the right behavior. You again get the xylem axis, the thing driven by auxin. There's the expression of the auxin response gene. And there's the localization of AHP as observed in the GFPs.
A question?
Yep.
You said the gradient of this microRNA is radial.
Mm-hmm.
So why doesn't it inhibit the AHP at the ends of the pole?
PHP is suppressing AHP. So the microRNA level is dropping as you go in. That means the PHP level is going up because there's less microRNA to remove it. So in the central part there's high PHP, and that's why this bit's blue. At the edges you've killed off the PHP. That makes sense?
I
Yeah.
thought the microRNA pattern would be symmetric.
It is, but remember originally the AHP wasn't symmetric, it was the line across the middle. So the thing we've done is knock out the circular bit in the middle. So the effect is radial, but the initial pattern wasn't, which is why this isn't. Okay. I want to say anything else about that. Oh yeah, incidentally, so P is the protein and M is the mRNA. So occasionally we plot both. So we do the usual thing to test whether the model is making a bit more biological sense than just testing it against the known stuff. So the usual thing is to mutate things. So this is removing short root, so doing a short root mutant, which means you get rid of the PHP, which means, right, this is where I get muddled. Which way around is this? I don't know whether to give up. So the right, it's partly because you've indirectly confused me now.
Well, I'm confused too, so.
So the PHP takes down the trouble is there are so many arrows in this, I always get confused. The PHP takes down the AHP. So the PHP is present everywhere, so the AHP is also present everywhere. No, the AHP is not present everywhere, I got it the wrong way round, which means auxin is not having the effect. Right, so cytokinin dominates, and the pins are in a wider range than before. The only place cytokinin doesn't dominate is where we put in the repressor. Okay, and correspondingly that's the pin 7 GFP, so that's everywhere, it's all the way across what would have been the xylem axis. And correspondingly the auxin response is low, as it is here, it's just localized in the middle.
Sorry, when you say put in, that means initial conditions.
Tell me what I just said.
You said you put in kin.
Yeah, so right, so no, this is passive, this is just stuck down there.
Okay.
Right. Yeah.
It means
Yeah,
different type of cells.
That's an unknown signal, and therefore we don't let it evolve. To say that's the thing I like least about this earlier part of the work. Okay. So that's my first set of statements about pattern forming. We got a situation now where a pattern can be sustained. So if you perturb slightly away from it, it's driven back to that state. So one would hope that in a growing root, that's how the pattern is sustained. The question is, where does it come from in the first instance? In particular, why are there these two different poles, not more? Why is it an axis rather than some other structure? So the simplest conjecture for the two poles is it's to do with how the thing develops from embryo. So this is an AHP signal in the early growth of Arabidopsis, and it does have this bilateral symmetry for AHP. So I need to say slightly more about different plant types in a second. This is a slide I'm not going to go into details of. This is a related model by a different group who do propose that it's, I partly put it up to indicate that other people are working on similar things, but also to say that, as I understand it, the proposal in this paper is the mechanism for that bilateral symmetry is that the embryo itself has the bilateral symmetry. So I need to go slightly beyond that. So the plant biologists cunningly subdivide plant species into two. So based on the cotyledon is essentially the embryonic leaf. So monocots have one leaf. I'll leave you to speculate what dicots have. So Arabidopsis is a dicot, and that's what was on the slide two slides ago. So it does have something that mimics the two-pole structure we've seen of AHP. But that certainly can't be the whole story, right? So there is this simple correlation in Arabidopsis, but when you start to look at other species, it doesn't hold up. So here's another dicot, but it's got 4 xylem poles. They're rather hard to see. Rice is a monocot, but it has 6, which you can see around there. So Tony had, I think, a project student just doing some kind of routine stuff on counting number of poles against the size of the cross-sectional size of the plant root, and there is a pretty good straight line. So the next set of things I'll say is based on the hypothesis that it's the size of the cross-section that determines the pole number rather than the number of cotyledons.
Is there anything special about having 2 poles? Because if you look at that one bar, it's always, it's everywhere basically.
Yeah. I mean, arguably, there certainly is a correlation with size, right? I mean, I guess if I wanted to sell our model, I would argue that the kind of, you'll see 2 poles appearing again in different size roots later, but you'll see other stuff. I guess the simplest symmetry break would be to two poles, wouldn't it? Yeah. Okay, so further evidence for this. This is this cytokine in mutant again, wooden leg. So in the mutant case, you just, because cytokine is not operating, you see auxin everywhere, but you can rescue it by mechanisms I don't exactly understand. But so this is a route you take from having purely xylem. You rescue the cytokinin response, let the thing continue to grow, and the 2 poles indeed form, which is further evidence that the thing is not driven by signals from the shoot. There's something else going on in the root itself that's driving the morphology. Okay, so remember I said when we did the simulations I've already shown, it was based on sticking the pin 7s initially in the places where they're observed to be. This is simulations based on other initial data on the pin level, so exactly the same model, but initial data. So the pattern isn't robust in that sense. You're seeing something akin to xylem poles, but you're seeing, if you like, other poles as well, and they're not even evenly spaced, I guess, and that's the contrast from the observations. So one way of getting things to be more robust is to use timing. So this is, and again, I think there's limited experimental evidence that makes sense of this, that the cytokinin response is slower than the auxin one. So we switch on the auxin response and drive that to steady state, and then ramp up the level of cytokinin. So this is, if you like, the time course. And you go from a state, again, not robust at all, you limit down the number of poles, and you do get to a situation where you get the 2 poles we've seen in the earlier simulations. And then if you drive it up even further, not surprisingly, cytokinin wins everywhere. You've ended up with 1 pole here and then it'll disappear altogether. So that would be an argument that if you ramp up the cytokinin to some particular level, you're making the pattern more robust. But so far those pictures are all with exactly the same geometry I had before. We're now going to try and to repeat, this is all very preliminary, try and understand what happens if you make the root cross-section larger, so different plant species. And the way we've driven this as a computational experiment is to start off essentially with a single pole, so think of it as being driven from a monocot, have some initial level of auxin at this location, and then drive the simulations to steady state with the initial auxin level varying. So at low levels of auxin, you don't break the radial symmetry. It goes to something pretty much as a radiosymmetric case. As you ramp it up, you decrease the number of poles. You get to state starting to look a bit more reminiscent of rice, so more xylem poles, arguably getting towards periodicity.
What is the time scale for reaching steady state?
No idea. Given we don't have realistic estimates. If that's a biological question, well, I don't know the answer to the question whether it's biological or mathematical. If it's biological, I don't know. If it's mathematical, we don't have realistic values for all the parameters anyway. So, slightly undershot, but there's a few extra things to say by way of conclusion. So these are Nathan's viewpoints on the upshots of the model. So we do have, this isn't a conclusion listed here, the initial conclusion is the model as it stands is capable of reproducing the patterning that was observed, as long as we start sufficiently close to it. When you move away from that and start to think about pattern robustness, there does seem to be some relationship between the size of the steel and the number of poles, which is at least not inconsistent with the experimental evidence. And this is the obvious rider that we need to do far more work to understand the dependence of the vasculature structure on the cell geometries, on the initial data and so forth. There's a final couple of slides on work in progress, which does go back a bit more to the issue of general mathematical questions rather than biological specificity. So this is what Nathan's doing at the moment, which is very much in the spirit of what Turing did originally. Actually, I don't think Nathan knows that. So this is the schematic so he's doing cells in a ring, so identical cells. He's just shaded them differently, and he's got more cells than in that picture. And then he's letting it evolve, and I think this is the auxin levels. So there's indications of some relatively interesting dynamics. It sort of settles down to a metastable uniform state, and then bifurcates off into localized poles, and there does look to be some kind of phase coursening going on. So I think there's some interesting mathematics of pattern formation underlying this, because the mechanisms for driving the pattern are not the familiar ones of Turing and so forth. But we haven't yet done much on that. And the other thing to say is, of course, plant roots are three-dimensional. So the stuff, they are long and thin, so the two-dimensional cross-section is a sensible thing to do. But the kind of patterns I've talked about really focus on the initial formation of the xylem. There's current work going on taking three-dimensional geometries and doing the corresponding computational analyses and focusing on the various different pin types that vary, as indicated in the earlier picture. And finally, the people involved are listed there. So Nathan and Daniele did most of the modelling work. Leah's another modeller at CPIB who was also involved. Tony leads the experimental team at Nottingham, so these are his postgrads and postdocs. There's collaborations with Sainsbury here, so I should say the experimental pictures, I don't guarantee I know who did each of them. So these are experimental groups as well. Tony was at Helsinki for a chunk of time, and he's also collaborating with Sainsbury here. And I'm done with that. Thank you.
Thank you very much. Yeah, we have time for questions. Peter?
I have 3 questions.
Mm-hmm.
One of you, can you recover a continuum model?
I don't think it's a sensible thing to do.
Okay.
Because I can show you any picture. The trouble is, right, so neighbouring cells are different, right? So the patterns you're trying to form are on the cell scale. So you might get some insight by writing down a continuum model. I think you're better off sticking to discrete, to be honest. Goes against the grain for me to say that, but yeah.
What about the boundary conditions?
So on the edge of the tissue, nothing gets out. I imagine that's what's been done.
Right. I think you spoke about growth. The last picture you showed that you claim maybe it might not be Turing model for the cycle. You have just 3 slides.
This one?
No.
That one?
Yeah, that one. So what you're observing on the right-hand side,
Mm-hmm.
we routinely observe these for growing patterns, growing domain.
Right, but this isn't a growing domain. The reason this slide is misleading is there's far more cells there than there are there.
That's right.
Yeah.
So in your model, you don't have growth. I mean, is growth important for you?
No, not in this part of the root. Sorry, I have to go right back to the first picture. So the stem cells and stuff are down here. So there's the cell division here. There's the elongation zone here where the things are expanding. I think. Do I go into this? This is maybe in the elongation zone. Shows how dodgy I am on plant biology. It may still be growing in the vertical direction in the region we're interested in, but it's not growing radially.
Yep.
So in terms of the cross section, it's not an issue.
But you're interested in the radial, I guess.
For this purpose, we are. Other stuff we're doing does have growth and mechanics in it, right? Yeah.
I don't fully understand what you were doing, but is it that you had a system of ordinary differential equations for lots of components, and there were some of them were associated with each cell. Each cell has a list, and there's a possibility of a flux from one cell to another. So from that perspective, you could think, and because the cells are arranged in this kind of geometrical pattern, like rings, so I guess there is, suppose there could be some kind of discrete geometrical equations in some sense. What could it? I don't know. Because when I saw your results seem to be lit up in rings.
Yeah. So
Kind of I don't know whether you think that there is some process whereby patterns move around in a ring dynamically, or whether there's interaction between one ring and the adjacent rings.
So the patterns I've shown are all static. There is interaction between the different rings. I don't know what I'm looking for here. So there is transport between neighbouring cells in all directions, right? But some of it directed by the pin carriers.
How many rings? If you take one ring, how many cells might you have in a ring?
I can probably count them. 1, 2, 3, 4,
Oh, that's
5,
just
6, 7, 8.
very small.
Yeah.
That's it.
It's that. I mean, that comes back to Anatolia's question. There's very definitely a finite number of cells in there.
And a small number.
Yeah.
So essentially, the simulation of this is solving ordinary differential equations.
Yeah.
But in the interesting question, is there any mathematical structure in those ordinary differential equations which could give some useful?
Yeah, I mean, I think the most interesting bit is the transport. So there's all sorts of things that potentially will be done because, as always, there's multiple timescales and things in here. But the transport isn't completely standard. Yeah, so there is this feedback between the level of the proteins in this cell and how fast stuff gets transported to the next one, which in turn feeds back into the gene regulatory processes. So this reflects actually quite a complex feedback loop.
There where you could use neural networks, because the structure is probably very similar. So where you try to learn how these particular patterns are connected. Basically, you would like to learn the fluxes and probably some of the kinetic constants.
Yeah, so there's a pile of stuff in principle to be done in terms of parameterization and so on, but one would need more data from that. We are focusing on bottom up because Tony in particular is doing a pile of stuff in terms of identifying the relevant proteins. So we're trying to mechanistically feed in the biological knowledge. I don't know how that relates to your question, but that's kind of the philosophy. Yeah.
The auxin pin dynamic, I mean the interaction between auxin and pin 7, is it similar to what people are talking about between auxin and pin 1?
So as will be clear, that in the model there's no interaction at all between, cytokinin drives pin 7.
I mean, according to what you see, I mean, pin is polarized, right?
Yeah.
7. And because it's shown that auxin pin dynamic, if you can easily develop some models with some interaction between pin 1 and auxin that is very nicely generates this kind of patterns, and it's tunable. And
right, you're right that there's a pile of stuff on coupled models of auxin transport with pin levels. As far as I know, there's no evidence that happens with pin 7. I may be wrong there, but it is known that pin 7 is regulated by cytokinin. So in the sense of minimal modelling, this is the place to start.
And you haven't been thinking about mechanical signals at all?
Not in this context, because it's not, at least in the radial sense, it's not in the growth zone. To repeat, in other contexts, in the stuff lower down we have.
Those models that have been shown that stress is able to be employed to regulate the pin level.
I don't, so for example, we're doing stuff on lateral root formation, which I think that may well be relevant to. I suspect it's not relevant here.
Okay. Thanks again, John.
Okay.